Have you ever wondered how scientists detect and identify the specific protein of interest from a complex mixture of proteins or a sample? A complex world hides behind this simple question, and today we are going to unravel one part of it that is called Western blotting. There are different types of blotting: southern blotting, western blotting, and northern blotting. But here we are talking about western blotting, as blotting is a technique for detecting macromolecules. If you are trying to detect DNA, we use southern blotting. If you are trying to detect RNA, we use northern blotting. But if we want to detect proteins, we use Western blotting. Western blotting is a very sensitive type of the technique that can even detect nanograms of the protein molecules in mixture of the other proteins. Western blotting is not only a technique that detects the specific protein but also used to detect the chemical reactions going on inside the cell, as well as to find the total proteome in the cell. Helps to understand about protein-protein interaction. A typical Western blot technique includes the following steps: separation by size, transfer of protein to a solid support, and detecting the target protein using antibodies to separate macromolecules in a sample. The first step in the STS page procedure is a sample preparation, so that a loading buffer containing STS, beta-marcaptoethanol, bromophenol blue, and glycerol is added to the sample proteins. The loading buffer is used in order to give all proteins present a uniform negative charge. Since proteins can be positively, negatively, or neutrally charged, once the loading buffer is added, the samples are then heated to 95 degrees Celsius. Proteins are large biomolecules consisting of one or more long chains of amino acid residues. They are formed by linking amino acids with peptide bonds and folds into the specific spatial conformations driven by a number of interactions such as hydrogen bonds, hydrophobic interactions, disulfide bonds, and ionic bonds. STS is an anionic surfactant that contains a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. STS denatures the native proteins by disturbing the non-covalent forces including hydrogen bonding, hydrophobic, and ionic interactions, while the reducing agent beta-marcaptoethanol is used to cleave the disulfide bonds. Also, STS binds fairly uniformly to the protein, and intrinsic charges of this protein become negligible when compared to the negative charges contributed by STS. This treatment brings the folded proteins down to the linear molecule with net negative charges. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. The gel is produced by polymerization between the two glass plates anchored vertically in a cassette. The gel cassette is placed vertically between the two electrodes, positive electrode located at the bottom of the gel, whereas the negative electrode is positioned at the top of the gel. Next, the gel is inserted into the chamber. Then a running buffer is poured to allow the conduction of the current through the gel. SDS is also present in a gel and in a running buffer, to make sure that once the proteins are denatured, they stay that way throughout the run. During separation, a molecular weight size markers is usually loaded onto the gel. This consists of the protein of known sizes and thereby allows the estimation of the sizes of the protein in the actual samples, which migrates in a parallel in different tracks of the gel. Each sample is added into its own well in the gel. As we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, while glycerol increases the density of a sample and it is used to fold the sample into the well. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel, away from the negative electrode and towards the positive electrodes. Also, small proteins migrate relatively easy through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly. Due to the relatively small molecular sizes of the bromophenol blue, it migrates faster than proteins, and by optical control of the migrating colored bands, the electrophoresis can be stopped before the samples have completely migrated through the gel and leave it. Once the proteins are separated, the gel cassette is removed from the electrophoresis tank. Next, the glass plates are removed, and the top portion containing the wells is cut off from the gel. In the next step, the separated proteins will be transferred from inside the gel to an appropriate membrane. When performing a wet transfer, the gel is first equilibrated in transfer buffer so that it is placed in a tray with the blotting buffer on a rocking platform. To make a blotting sandwich, a gel holder cassette is immersed in the transfer buffer with the black side down and the white side up, and out of the buffer. Next, a fiber foam pad is immersed in the transfer buffer. Then it is laid on the black side of the gel holder cassette. After that, a piece of a filter paper is soaked in the transfer buffer, and then it is placed on the top of the fiber pad. Once the gel is being equilibrated in the transfer buffer, it is carefully placed on the blotting paper. For the transfer, a nitrocellulose membrane or a polyvinylidene difluoride membrane can be used. The membrane is soaked in the transfer buffer, then it is placed squarely on the gel. Next, a second sheet of the blotting paper is wetted with the transfer buffer. Then it is placed on the top of the membrane. Finally, a second fiber pad is immersed in the transfer buffer. Then it is laid on the top of the blotting paper. Next, the gel holder cassette is closed and locked with a clamp. The most commonly used method for transferring the proteins is called electroblotting. During this transfer, an electrode assembly with positive electrodes and negative electrode is used. The gel holder cassette is inserted into the module, ensuring that the cassette is properly positioned from negative to positive. The inner module is placed into the electrophoresis chamber, then the chamber is filled with the blotting buffer. Next, a lid is placed onto the electrophoresis tank and electrical LEDs are connected to the power supply to run the blot. Electroblotting uses an electric current to pull the negatively charged proteins from the gel toward the positively charged electrodes and into the membrane. The proteins move from within the gel onto the membrane while maintaining the organization they had within the gel. When the run is complete, the electrophoresis chamber is disassembled and the inner module is removed. Then the gel holder cassette is removed and opened. Next, starting with the fiber pad, each layer is removed until reaching the membrane. After the electrotransfer of the proteins on the membrane, the next step is the detection of our protein of interest using antibodies. First of all, the membrane is soaked in a blocking solution which contains bovine serum albumin. This step must be taken to prevent the interactions between the membrane and the antibody used for the detection of the target protein. Once the blocking solution has been added, the membrane is incubated on the rocking platform. The BSA protein attaches to the membrane in all places where the target proteins have not attached. Thus, when the antibody is added, it cannot bind to the membrane, and therefore the only available binding site is the specific target protein. After the incubation is complete, the blocking solution is removed. Then a solution containing the primary antibody is poured into the tray. Next, the membrane is incubated under gentle agitation. The primary antibody binds to the specific target protein. Following incubation, the primary antibody solution is removed, then a wash buffer is poured. Next, the membrane is incubated on the rocking platform. The membrane is washed several times in wash buffer to remove unbound primary antibodies and thereby minimize background. After washing the membrane, the wash buffer is removed, then a solution containing the secondary antibody is added into the tank. Next, membrane is incubated under gentle agitation. The secondary antibody recognizes and binds to the specific portion of the primary antibody. Following incubation, the secondary antibody solution is removed, then the wash buffer is poured into the tank. Next, the membrane is incubated on the rocking platform. The membrane is washed several times in wash buffer to remove unbound secondary antibodies. It is crucial to thoroughly wash the membrane at this step. After rinsing the membrane to remove unbound secondary antibody, the membrane is incubated in a solution containing the substrate. Chemiluminescent Western blot detection is the most frequently used method to detect the proteins on the Western blot. To allow the detection of the target protein, the secondary antibody is commonly linked to a reporter enzyme such as horseradish peroxidase. The most popular Western blot substrate are luminol based in the presence of a hydrogen peroxide. Horseradish peroxidase enzyme catalyzes the oxidation of luminol to excited state product called 3-amino phthalate. This product decays to a lower energy state by releasing photons of light at 425 nanometer. The production of luminescence is proportional to the amount of horseradish peroxidase conjugated to secondary antibody and therefore indirectly measures the presence of the target protein. After incubation, the membrane is placed in a clear plastic wrap to prevent drying. The membrane is analyzed by densitometry, where the resulting light signal is detected by CCD camera, which captures a digital image of the Western blot. According to the obtained bands, we can determine the approximate size of our protein of interest as well as its presence or absence in each sample.